DNA footprints. How scientists track unknown sources without direct contact. Imagine identifying a person who was never photographed, never recorded, never touched, and may not even know they left behind evidence. No fingerprints, no cameras, no witnesses, just invisible molecular traces drifting through air, water, or soil. This is not science fiction. This is modern environmental DNA, forensic genomics, and probabilistic genetic tracking, a rapidly evolving field where biology meets machine intelligence. Today, DNA footprints allow scientists to detect organisms, reconstruct movements, identify unknown sources, and even predict biological presence without ever making direct physical contact. At its core, this technology reads the faint molecular echoes every living system leaves behind. Every organism continuously sheds genetic material. Humans alone release roughly 40,000 skin cells per minute, each containing complete DNA. Hair fragments, saliva aerosols, sweat molecules, respiratory droplets, and microscopic tissue particles form an invisible genetic cloud around us. These fragments degrade quickly. UV radiation, oxidation, bacteria, and temperature break DNA into pieces within hours or days, yet even partial sequences are enough for identification. What scientists analyze today is rarely whole DNA strands. Instead, they recover fragmented nucleotide chains often shorter than 100 base pairs, then computationally rebuild probabilistic matches using genomic reference libraries. The foundation of DNA tracking begins with polymerase chain reaction, or PCR, a molecular amplification process capable of multiplying tiny DNA fragments billions of times within hours. A single molecule can be copied into millions. PCR operates through thermal cycling, heating DNA to 95 A circumflex degree C to separate strands, cooling to 50-65 A circumflex degree C for primer binding, then raising to 72 A circumflex degree C for enzymatic replication. Each cycle doubles the DNA. After 30 cycles, one molecule becomes over 1 billion copies. This exponential amplification allows scientists to detect genetic material present in concentrations as low as 1 femtogram, roughly 1/trillionth of a gram. But amplification alone is useless without interpretation. That's where sequencing enters. Modern laboratories use next-generation sequencing platforms capable of reading billions of DNA fragments simultaneously. Machines like Illumina, Novasec, or Oxford Nanopore systems convert molecular sequences into digital data streams, translating adenine, thymine, cytosine, and guanine into binary information. A single environmental sample such as river water or air filter dust may generate terabytes of genomic data. Artificial intelligence then sorts these fragments, compares them against reference genomes, and assigns statistical probabilities to organism presence. This approach is called environmental DNA, or eDNA. Instead of capturing animals or people, scientists analyze water, soil, air, or surfaces. 1 liter of lake water can reveal hundreds of species. A cubic meter of air contains enough biological material to identify mammals, birds, insects, plants, fungi, and bacteria. In controlled studies, researchers detected human DNA from empty rooms hours after occupants had left. Even footprints on sand leave trace genetic signatures that persist for days depending on humidity and UV exposure. What makes DNA footprints revolutionary is that they do not require knowing what you're looking for. Traditional forensic science works backwards from suspects. DNA footprinting works forward from environments. Algorithms scan samples against massive genomic databases, identifying organisms statistically rather than directly. This transforms biology into a passive sensing system like radar, but molecular. Tracking unknown sources becomes possible because DNA contains population markers. Certain genetic variations, single nucleotide polymorphisms or SNPs, correlate with geographic ancestry. By analyzing hundreds of SNPs, scientists can infer continental origin with over 95% accuracy. Larger data sets enable regional prediction. Combined with isotopic signatures in hair or bone such as ratios of oxygen 18 to oxygen 16, researchers can estimate where someone lived, what climate they experienced, and even aspects of their diet. This is no longer limited to humans. Wildlife researchers use DNA footprints to monitor endangered species without disturbing habitats. Snow samples reveal polar bear movement. River water exposes migratory fish paths. Forest air identifies elusive mammals. In some conservation studies, eDNA detected species missed entirely by camera traps. Detection sensitivity often exceeds 90%. compared to traditional survey methods averaging 40-60%. Now add machine learning. Modern genomic analysis pipelines use neural networks trained on millions of sequences. These systems recognize mutation patterns, classify unknown fragments, and reconstruct partial genomes. Deep learning models can predict organism identity even when only 0.1% of the genome is present. Bayesian inference assigns confidence scores, enabling probabilistic tracking rather than absolute identification. This matters because real-world DNA samples are messy, degraded, mixed, contaminated, incomplete. AI thrives in uncertainty. Consider airborne DNA. Recent experiments used high-volume air samplers drawing 300 liters per minute through filters. DNA recovered from urban air contained traces of dogs, cats, rats, birds, plants, and humans. In controlled zoo environments, researchers identified every animal species in enclosures using only air samples. This opens possibilities for remote biodiversity mapping, epidemiological monitoring, and ecological surveillance, all without direct observation. DNA footprints also interact with physics. Molecular dispersion follows fluid dynamics. Airborne DNA behaves like aerosolized particles governed by Navier-Stokes equations. Turbulence, humidity, temperature gradients, and airflow determine how genetic material spreads. In water, DNA diffusion is affected by viscosity, currents, and microbial degradation. Models estimate DNA decay half-lives ranging from 3 hours to several days, depending on environment. These decay curves allow scientists to infer how recently an organism passed through an area. Time becomes measurable. If DNA concentration drops exponentially according to CT equals C0 A tilde, E a circumflex letter to C6KT, where CT is concentration over time, K is decay constant, and C0 is original concentration. Researchers can back calculate approximate presence windows. This transforms DNA into a biological timestamp. Now scale this globally. Satellite-assisted ecological genomics integrates DNA sampling with Earth observation systems. Multispectral satellites track vegetation, temperature, moisture, and land use. When combined with ground-based eDNA sensors, AI models correlate genomic data with environmental conditions. This allows predictive mapping of species migration, invasive spread, and ecosystem collapse. Some experimental platforms integrate genomic sensors with autonomous drones, creating mobile biological monitoring networks. Even more fascinating is genetic shadowing. Every environment carries layered DNA histories, present organisms overlaying remnants of past activity. Soil cores contain centuries-old genetic material. Ice samples reveal ancient ecosystems. Deep sequencing can reconstruct long-lost biodiversity. In Siberian permafrost, scientists recovered DNA over 2 million years old. These molecular archives allow reconstruction of evolutionary timelines with unprecedented resolution. But DNA footprints also raise ethical challenges. Because genetic traces are unavoidable, privacy becomes complex. You cannot stop shedding DNA. Your biological identity exists in every room you enter. This forces society to rethink consent in a world where molecular data is ambient. Current regulations restrict forensic use, but technological capability advances faster than law. The same tools that protect wildlife can potentially track humans. This dual-use nature demands strict governance. From a scientific perspective, however, DNA footprinting represents a paradigm shift. We no longer need to see organisms to know they exist. Biology becomes detectable at distance. Life becomes readable through environments. The planet itself turns into a massive genomic sensor. Future systems may integrate quantum biosensors capable of detecting DNA without amplification. Nanopore arrays embedded in infrastructure could continuously scan air and water. Edge AI devices might process genetic data locally in real time. Synthetic biology could create programmable organisms that act as environmental reporters. These ideas sound futuristic, but prototypes already exist in laboratories. Imagine cities where invisible genetic currents are mapped like weather. Forests monitored continuously for biodiversity loss. Oceans surveyed molecularly for ecosystem health. Disease outbreaks detected before symptoms appear. This is not speculative warfare technology. This is civilian science reshaping environmental awareness, medicine, and conservation. DNA footprints teach us something profound. Every living being writes itself into the environment. Every movement leaves molecular echoes. Life does not pass silently through space, it records itself continuously. And we are just learning how to read it. As sequencing costs drop from $100 million per genome in 2001 to under $200 today, access expands globally. Developing regions can deploy genomic monitoring for agriculture, water safety, and public health. Citizen science projects already use eDNA kits to map local biodiversity. The molecular future is becoming democratized, but the deepest impact may be philosophical. DNA footprinting reveals that individuality is temporary, but biological presence is persistent. We are not isolated entities. We are molecular participants in planetary systems. Our genetics mingle with air, soil, water, and life itself. The invisible world is not empty. It is alive with information, and science has only begun decoding it. Strong curiosity ending. If DNA can tell us who passed through a forest, who swam through an ocean, who breathed inside a room, then the next question becomes unavoidable. What else is the environment already recording about us? And more importantly, who will learn how to read it first? If you want the next episode where we explore how AI reconstructs entire ecosystems from microscopic traces, subscribe now. Because the future of science isn't happening in laboratories anymore. It's happening invisibly all around you.